Hi, I'm Jeff Mogridge and I'm going to give you an introduction to the Chemical Engineering and Biotechnology Tripos. So the first thing to say is that the Tripos is a three-legged stool on which students sat to take their exams a few hundred years ago. Now it just means course, so I'm going to tell you about the Chemical Engineering and Biotechnology course at Cambridge. The second important point is that Chemical Engineering and Biotechnology is a single course. We're going to teach you both subjects. The course is designed to produce graduates that meet the needs of the modern process industries, and that means technical competence, personal and transferable skills, a strong emphasis on fundamental principles used to solve practical problems for processes and for products, and an ability to cope with modern scientific advances. The key thing about our course is that it's both interesting and it's useful. So the structure of the course, it's a four-year course. You come in the first year and you take what's called CEBT 1A. This is fundamentally underpinning science which you'll need to solve the problems of engineering later in the course. You do chemistry and maths with the first year natural science students, and you'll also do subjects within the chemical engineering department, including sustainability, and also a mechanical design and manufacturing course. This involves CAD drawing a device to solve a particular problem, and then using techniques such as 3D printing, laser cutting, CNC machining and so on to make that device. Year 2, 1B, is core applications. So this is where you really start to use the scientific knowledge to solve engineering problems. In year 3, you continue with those core applications, but there's also a major design project, and that involves working in teams of around 6 and designing a piece of chemical engineering or biotechnology equipment, possibly a whole chemical plant. Now, it is possible to leave the course after 3 years, and in that case you would graduate with a BA. The great majority of our students stay on to complete the 4-year course, and many of them will do some vacation work between years 3 and 4 to give themselves experience in industry. The 4th year, part 3, is much more optional work. So years 1 to 3, everybody does the same courses. In year 4, you have quite a bit of choice. You also do an advanced design project and an extensive research project accounting for about half of the credits in the final year. So each student works in usually a pair to tackle a novel research problem supervised by one of the academics in the department. If you stay for the 4 years, you graduate with a BA and an MEng degree. So the course is taught by a number of different elements. So the syllabus is essentially set by the lecture courses. That involves one lecturer talking to, say, 40 or 50 students. And typically you'd have 8 to 10 lectures per week. We also provide a substantial amount of practical work. I've already mentioned the workshop experience you'll get. We also have practical classes which usually involve work for 1 afternoon per fortnight in the first 2 years of the course. You write reports on those lab classes, and those go towards your degree as assessed credit. We also have a substantial amount of non-practical project work. This involves often working in teams and tackling a problem which is more extensive and more difficult than it would be possible to do in an exam. So, for example, it might require extensive computational work. Typically you'll solve one of these more extensive problems one every 3 or 4 weeks in the first 3 years of the course, and there's also the main design project that I mentioned in year 3, which is also a piece of project work. That could be design of a chemical or biochemical plant and runs over 6 to 8 weeks. Again, the project work forms part of the assessment. Overall, the practical and project work continual assessment accounts for about 1/3 of each year's mark, the other 2/3 coming from exams. In addition to the teaching provided by the department, in Cambridge the colleges provide small group supervisions. and that involves groups of 2 to 3 students discussing problems and the lectures with a supervisor who's an expert in the field. That may be a PhD student, it may be a postdoc, or one of the lecturers who gives the lectures that's being discussed. The colleges also provide pastoral support, libraries, sports facilities, accommodation, food and so on. So moving on from the course itself, I'd like to tell you a little bit about what it is that chemical engineers and biotechnologists do. Essentially they design and operate processes to make chemical or biochemical products. And the key thing is that they do this on a large scale, and increasingly there's emphasis not only on efficiency but also on safety and cleanliness, and that of course includes sustainability and reducing the carbon footprint of any of these operations. When we look more specifically at what you might do, actually it's extremely varied. So for example, one of my students went on to design the small Pringles tin. Another ran the largest sewage plant in Egypt, and another started a chocolate factory in Poland. So there's a huge variety of what our graduates go on to do beyond Cambridge. Let me make a few generalisations about engineers and chemical engineers and biotechnologists. When one thinks about scientists, what you're really thinking about is people who make new discoveries and acquire novel knowledge. Many of those discoveries, of course, become useful in the real world. And that's where the engineers come in. Engineers apply the science to design and build things. And those things need to be useful in the real world. So those might be engines, machines, structures, electrical equipment, or in our case, chemical or biochemical products. Now, I think it's the case that scientists are often seen as more glamorous than engineers. They certainly appear on the news more often, I think. But it's definitely the case that society needs a lot more engineers than it needs scientists. There's more need to apply the science than there is to discover new science. And what do chemical engineers and biotechnologists do? They're distinguished by the fact that they work on processes which involve some form of chemical or biochemical change. So they're engineers who work on chemical and biochemical change. Now, just to give you a few examples of people who've studied chemical engineering in the past, this is Johnny von Neumann. He was generally reckoned to be the greatest mathematician of the 20th century, famous for very many things, but in particular starting the US digital computer program. This is Margaret Hutchison Rousseau. She invented what was called deep tank fermentation, and this allowed penicillin to be manufactured at large scale in time for the D-Day landings in 1944. So instead of being grown on little Petri dishes, she allowed penicillin to be manufactured in 10,000 litre fermentation reactors. And that resulted in 2.3 million doses of penicillin being ready for the Normandy landings, and that's reckoned to have saved the lives of 12 to 15% of the people who participated in those landings. She was also the first woman in the US to get a doctorate in chemical engineering and the first woman member of the American Institute of Chemical Engineers. This is His Excellency Professor George Maxwell Richards, the 4th President of Trinidad and Tobago. He was actually a graduate of this department. This is Cindy Crawford, a well-known model and actress, and this is Jim Ratcliffe, who before he moved to Monaco, I think was the richest person in the UK. He's the chairman and chief executive of the chemical company INEOS, and also the owner of the cycling and America's Cup teams of the same name. So again, you can see there's a huge variety of what you can do if you study chemical engineering. And let's not forget biotechnology. This is Dr. Karina Joe. You've probably heard of Sarah Gilbert and Kath Green, who are the scientists who invented the Oxford vaccine. Now, obviously, that was an incredible achievement, but actually they only made 700 doses. And by the end of 2021, there'd been 2.4 billion doses administered in 172 countries. And Dr. Karina Joe was key to the scale up of the process from 700 doses to 2.4 billion doses. In January of 2020, she was the only person in Oxford working on the scale up of the manufacturing method. And that was absolutely key to the success of the Oxford-AstraZeneca vaccine. So the method used for purification of the vaccine in the lab involved what's called density gradient centrifugation, where you create a gradient of salt density in a tube, and your substances separate out according to their density in layers. And in order to achieve that, you centrifuge the tube at 154,000 times the force due to gravity for 2 hours. To give you a feeling of just how much force that is, astronauts will experience approximately 3 times the force due to gravity at takeoff, and fighter pilots can put up with about 9 times the force due to gravity for a couple of seconds. This is a completely impractical method to make millions of doses of the vaccine. So what Karina Jo did, amongst other things, was develop a purification process which involved ion exchange chromatography to replace that process. And that's just like the process you use on a domestic water softener. Many of you may have them at home. So what I think that example illustrates is that one of the key things about chemical engineering and biotechnology is that you have to work on an industrial scale. Scale up is a particularly important feature of the subject. So many processes are well established, possibly even easy to do in a test tube scale, but they can be much more difficult to achieve on an industrial scale. So let me give you an example. I think probably most of you could make a cup of tea without too much difficulty. But what about if I asked you to make 2 million cups of tea a day? And moreover, I want you to do it without wasting any energy, wasting any ingredients, or leaving any pollution behind, and minimizing the carbon footprint of those 2 million cups of tea. Clearly, that's a much more difficult problem. Now, I've just given you one example of scale-up from the Oxford-AstraZeneca vaccine case. I'm now going to talk about another one. This is actually a very old one, but is of critical importance not only at present, but also going forwards. It's ammonia synthesis. I'm sure that most of you will have heard of the Haber-Bosch process, in which you combine nitrogen and hydrogen to produce ammonia. On the right here you see what's called a Prilling tower on Teesside. I'm quite fond of this photograph because I worked at that facility in 1998. So what you do is you drop liquid ammonium nitrate at the top of that tower, and by the time it gets to the bottom you're blowing hot air up the tower. By the time it gets to the bottom you end up with a solid pellet of ammonium nitrate, and that's much more easy to handle than would be a powder. And ammonium nitrate is explosive, so it's particularly important that it's easy to handle. Ammonium nitrate is the primary ingredient in fertilizers worldwide. So this reaction, the Haber-Bosch process, was invented by a chemist, Haber, at small scale. The person who developed the industrial scale process was a chemical engineer, Bosch. Both of them got Nobel Prizes. Now, there's approximately 180 million tonnes of ammonia produced per year. About 80% of that goes into fertiliser. Interestingly, under current circumstances, 8% of that is produced in Russia and 2% in the Ukraine. Only about 0.6% is currently produced in the UK. Now, you couldn't possibly feed the world's population without fertiliser, without ammonia. So almost certainly most of the nitrogen atoms in your body come from your food, but via the Haber process. Now, a modern ammonia plant is enormous. They can produce around 1 million tonnes of ammonia per plant. So if you think about that, that roughly means you need, let's say, 200 to 300 ammonia plants worldwide. Each country probably has 1 or 2. I think the UK has 2. Now, this reaction, I'm sure you've heard this before, is carried out at high pressure, 100 bar, high temperature, 450 degrees C, and requires an iron catalyst. And even with all of that, you only achieve around a 15% conversion in a single pass. What that means is that you need a recycle, and if you need a recycle, you need to separate out the product, ammonia, before you recycle the unreacted reactants. Now that is classical chemical engineering. So this is the sort of scheme you develop. You put nitrogen and hydrogen into the reactor. It reacts, 15% of it reacts to produce ammonia. You then separate out the ammonia. In this case, you do that cryoscopically. You cool it down, liquefy the ammonia, and then you recycle the nitrogen and hydrogen to the start again. And if you do that, you can raise your overall production from the 15% conversion up to 98% conversion of nitrogen and hydrogen. So where does the nitrogen and the hydrogen come from for this process, I hear you ask? Well, you get it from methane. Hydrogen you get from methane. So the first step is to do what's called steam reforming in the primary reformer. You treat methane with steam, and that produces carbon monoxide and 3 hydrogens. There's then what's called a secondary reformer, and that takes more methane, reacts it in this case with oxygen, and actually the oxygen comes from air, so along with each oxygen atom there's approximately 4 nitrogen atoms, and that produces again carbon monoxide and hydrogen, but of course it's mixed with nitrogen because you reacted with air in the first place. Now remember, we needed hydrogen and nitrogen for the Haber-Bosch process, and this is the source of the nitrogen for the process. We don't really want carbon monoxide hanging around, and also if you treat the carbon monoxide with steam in the water-gas shift reaction, you can make more hydrogen and carbon dioxide, which obviously is a much less hazardous byproduct than would be carbon monoxide. So if we multiply the first reaction by 3/8, the second one by 1/4, and then the third one by 7/8, add them all together, what we end up with is a reaction of methane, water, oxygen, nitrogen being present because the oxygen is provided in the air, and the product is 7/8 of a CO2 molecule, 3 hydrogens and 1 nitrogen. That means we've got 3 hydrogens and 1 nitrogen to put into the Haber-Bosch process, which is exactly the stoichiometry we need. And we've got 7/8 of a CO2, which is a byproduct, which we need to separate out. Some of you will remember over the summer that when the gas price went up a lot, the ammonia plants in the UK shut down. That means that the CO2 byproduct was not being produced, and that means that the food and drink industry ran out of CO2. And so this became a major problem, not only because we were no longer producing fertiliser, but because you couldn't make fizzy drinks, you couldn't preserve meat and vegetables and so on in CO2 gas. However, most of the CO2 doesn't go into the food industry, it goes into the atmosphere. So here's our overall reaction stoichiometry. If we work that out in terms of molecular weight, so the molecular weight of CO2 is 44, of ammonia is 17, that means that we're getting a little over a tonne of CO2 for each tonne of ammonia we produce by the Haber-Bosch process. And that's even if the process was perfectly efficient, which of course it isn't. There'll be some energy costs which produce more CO2. So more than 1 tonne of CO2 per tonne of ammonia. And remember I said there were 180 million tonnes of ammonia produced per year, so that's equivalent to more than 200 million tonnes of CO2 going into the atmosphere every year. And actually that turns out to be almost 2% of the total man-made CO2 emissions coming into the atmosphere. Now that's pretty close to the emissions from the whole of the aviation industry. I think the aviation industry is around 3% of total emissions. Now, of course, we could all fly less, but we can't all eat less. So there's not really a possibility of producing less ammonia unless we want food to become a great deal more expensive and people in the poorer parts of the world perhaps to start to suffer from malnutrition. So we don't want to produce less ammonia, but what we do want to do is produce it without the CO2 waste stream going into the atmosphere. And that's a big opportunity and a big challenge for chemical engineers going forward in this century. Now there's 2 immediate ways you might do this. So the first is that actually this is quite unusual in that what you've produced in this reaction is a pure carbon dioxide waste stream. And that's absolutely ideal if you want to do carbon capture and sequestration. So you take that waste stream and instead of sending it to the atmosphere, you bury it underground or under the sea somewhere, where it'll be stable for hopefully hundreds and thousands of years to come. So this is probably the best option to trial carbon capture and sequestration on a large scale in the coming decades. So there's one way of doing it. It'll add a bit of cost to the process, but at least we can continue to make fertiliser and grow food. The second option is actually to do away with the methane completely. So when Haber invented the process back in the early 20th century, he actually used electrolysis to produce hydrogen from water. So if you used that process, there would be no CO2 involved in the reaction. That would be a much more major change because you'd have to completely redesign the whole of the chemical plant. And of course, we've got, let's say, 200 or 300 of those very efficient chemical plants worldwide. That'll take much longer to do than would the carbon capture and sequestration. So this is a really nice example where chemical engineers have achieved a great deal over the last century, but also have a great deal of work to do in the coming decades to cut the CO2 out of the process. So what are we looking for from you if you want to apply for this course? Well, we want people who excel at and enjoy both maths and the sciences, but critically want to apply them to a real-world problem at large scale. So if you are going to obtain a place, you need, in general, to get 2 A stars and an A at A level, including maths and chemistry. And we have a preference for the third subject being another science, although that's not essential. If you're doing IB, that translates roughly to 40 to 42 points with 776 in the higher level, and again including maths and chemistry. The admissions process is actually handled by the colleges, and each college has slightly different requirements. So you should always check with the college you're applying to what their specific requirements are, but they will be pretty similar to those I've just outlined. Once you've applied, you'll have to sit the natural science admissions assessment in early November, and you may then be called to interview at the college in early December. Finally, if you would like some more information, I've provided some email addresses and websites down at the bottom of this slide. Thank you for attention, and I hope you'll be interested in applying to our course.